Hello everyone, welcome to the channel Study with Tasmita. Today we are going to start with the chapter number 1 of class 12, that is sexual reproduction in flowering plants. So in our previous class we have already classified all the living organisms on the basis of their similarities and dissimilarities. One of the hierarchical classification was kingdom. So plant kingdom was further classified into 5 divisions, which were thallophytes, bryophyte, pteridophyte, gymnosperm and angiosperms, right? So angiosperms are the most developed division of this kingdom as it bears flowers for its sexual reproduction. So we are going to take a detailed look about its sexual reproduction in this chapter. Before starting the chapter, we are going to discuss about the topic which we have to cover in this chapter, which are flower, a fascinating organ of angiosperm, then pre-fertilization, structure and events, then double fertilization, post-fertilization, apomixis and polyembryony. Now let's start with our new topic, that is flower, which is basically a fascinating organ of angiosperm whose origin is axillary bud. It is basically an organ which is a site of sexual reproduction in flowering plants. Before starting with the chapter, let us take a look of all the parts of a flower. If we talk about pistil or the carpel, it is the female part of a plant which consists of stigma, style and ovary. If we talk about the stamen, it is the male part of a plant which consists of anther and a filament. Now other these reproductive parts, there are accessory parts which are petal, which is a colorful part of a plant which helps in attracting the insects for pollination. If we talk about sepal, it is a green leafy like structure which provide protection to the birds in its initial stage. If we talk about the receptacles or the thalamus, it is basically a swollen part of a flower. Now pedicel, it is basically a part where bird is joined to the stem or a branch. Now ring-like structure formed by stamen is known as androecium, and group of pistil or carpel is known as gynoecium. Calyx, it is a group of sepals, and group of petals are known as corolla. Now let's start with another topic, that is pre-fertilization, structure and events. Now in the case of male, which is a stamen in a flowering plant, and it is a unit of androecium. So stamen is basically the male part, as we have discussed earlier, which consists of anther and the filament. Inside the anther there is a connective middle tissue. Anther, it is bilobed, means it has two lobes, right? And each lobe here two theca. So this is the diagram of theca here from inside. Okay, here is microsporangium in each theca, which consists of pollen sacs, one in each. Okay, basically it forms haploid pollen grains, or we can say two male gametes. If we talk about filament, this is the stalk-like structure which provide exposure to the anther. Now this proximal part is attached to thalamus. Now if we talk about connective tissue, it is a site of attachment of filament with anther. Now let's take a look at the structure of microsporangium, which consists of 4 walls and diploid sporogenous tissue. If we talk about the 4 walls, then out of these 4 walls, 3 walls are protective walls, and the fourth one, that is tapetum, is the nutritive wall. So the epidermis, endothelium, middle layers are the protective walls. Okay, epidermis and endothelium are the single layer, and middle layers are basically multiple layered, which is just next to endothelium. Now if we talk about the tapetum, which contains dense cytoplasm and are multinucleated. Now if we talk about the diploid sporogenous tissue, okay, which forms MMC, that is microspore mother cell, okay, that is also, then it further develop into pollen mother cell. By the process of meiosis, haploid microspores are formed, which further develops into pollen grains, which are also known as male gametophyte, which is 2 in number. Okay, so let us take a look of diagram. This middle part is the connective tissue, then this outer layer is the epidermis. Next to epidermis, it is endothelium, which are both are single layered. Okay, next to the endothelium, multi layered, which consists of multiple layers. Okay, tapetum is next to the middle layers, which provide nutrition to the growing pollen grain. Okay, and next to the tapetum, sporogenous tissues are present. Now if we talk about microsporogenesis, then it is the process of formation of haploid microspores from diploid microspores mother cell or the pollen mother cell, that is PMC, through meiotic division is called microsporogenesis. As we have seen, the sporogenous tissues present here, which consist of pollen mother cell, okay, in which meiotic division take place, and by the help of this division, haploid microspores are formed. This process is known as microsporogenesis. Then each microspore further develops to form mature pollen grain, that is male gametophyte. Now here in the diagram, the process of microsporogenesis is shown. Pollen grain represent the male gametophyte in the angiosperms. Okay. So the conclusion is pollen grain formed from microspores tetrad after microsporogenesis. Now coming to the topics, shedding of pollen grains on stigma. In 60% of angiosperm, pollen grain sets at two cell stage. One is generative and the other one is vegetative. Now in other 40% of angiosperm, pollen grain sets at three cell stage, that is one is vegetative, another two are two male gametes. Okay. Now we will talk about the harmful effects of the pollen grains. Pollen grains cause pollen allergy and also causes chronic respiratory disorder. The example of carrot grass, which is also known as parthenium or weed of weeds, which cause allergy, and in the case of respiratory disorder, it causes bronchitis and asthma. Now we are going to take a look of the importance of pollen grains. Okay, so pollen grains are basically rich in proteins. So in Western countries, it is taken as a food supplement in the form of tablets or syrups. Now if we talk about the viability of a pollen grain, it can be from 30 minutes to even several months and years, right? For example, in the case of wheat and rye, its viability is just for 30 minutes, and in the families such as Rosaceae, Leguminosae and Solanaceae, it can viable for several months and several years. Now pollen grains can be stored in liquid nitrogen at minus 196 degrees Celsius. It is very useful in plant breeding programs. Now with the next topic we are going to start with is the pistil megasporangium, that is ovule, and embryo sac, female gametophyte. Now here we are going to take a look of female parts and its function. Okay, so gynosium is basically is a group of carpel or pistil, okay, which consist of stigma, style, and ovary. So this is the stigma, this is upper flattened part, okay, this is the style, middle tubular part, and the ovary, this is the basal swollen part, which consists of one or more ovules. Now, in angiosperm, gynosium can be divided into two parts, that is monocarpellary, which the flower in which single carpel is present, okay, and another one is multicarpellary. It is further divided into two parts, that is fused carpels and free carpels. Flower with the free carpels are known as apocarpus and fused carpels are known as syncarpus. Now we are going to talk about the ovary, okay, which consists of ovary wall and ovarian cavity. Inside the ovarian cavity, locule is present, which is further divided into placenta and ovule. Placenta basically it provides nutrition to the developing ovule. Ovule is also known as megasporangium. Now we'll talk about the ovule or megasporangium. This is our topic. So it is followed. So its structure is divided into two parts, that is external and the internal. Funicle is the stalk of the ovule attached to the placenta. Then coming to hilum, attachment site of ovule with the funicle. Now integuments are the covering of ovule, which is further divided into two parts, outer integuments and inner integuments, okay. Now the micropyle end is a place where integuments are absent, which is just opposite to chalazal pole. Now chalazal pole is a part of ovule opposite to micropyle. Now if we talk about internal structure, it consists of nucleus, inside which diploid cells provide nutrition to the developing embryo sac. So micropyle is basically opening for ovule for the entry of pollen tube. Now let us take a look of its diagram. This is the funicle, okay. Next to it, it is the hilum. This is the outer integuments and the inner integuments. You can see here that it is the opening, which is known as micropyle, so that pollen tube can enter from here, and chalazal pole is just opposite to micropylar pole. Now this is the outer integuments, inner integuments, and these are the nucleus, and this is megaspore mother cell. Now we will talk about megasporogenesis, which is the process of formation of haploid megaspores from diploid megaspore mother cell by meiotic division. Okay, haploid megaspore develop to form haploid female gametophyte, that is embryo sac. Now this is the diploid megaspore mother cell. Now we can see here the diploid megaspore mother cell, okay, inside which nucleus is present, okay. Then it is followed by meiosis first. Now with the help of diagram, we are going to understand the process of megasporogenesis, okay. So now in the nucleus of megaspore mother cell, meiosis division takes place. The first meiosis first result into division of the nuclei, okay, and it forms megaspore dyad. Then again it divides and forms four nucleus in it, okay, out of which three degenerates and only one remains, the functional. In the functional megaspore, mitosis takes place and it forms two nucleate stage. Again it divides and forms four nucleate stage, which is followed by mitosis again, in which 8 nucleus are formed, okay. So this is the 8 nucleate stage. Here the cytokinesis take place. Okay, at the micropylar end, filiform apparatus, synergid cell, okay, and egg cell are formed. Right between at the middle, polar nuclei remain. Okay, this is only the one cell, and two nuclei are present here. Now, at the chalazal end, antipodal cells are formed, which is three in number. Three at the chalazal end and three number of cells at the micropylar end, and two nucleus, one cell is formed at the center. So this stage is known as cell and eight nucleate stage. Now, formation of embryo sac from single functional megaspore is known as monosporic. As we have seen above, that other three megaspores are getting degenerated, right? And only one of these are functional megaspore. So such kind of development of embryo is known as monosporic development. Okay. Now, if we talk about female gametophyte, that is embryo sac, also 7-celled and 8 nucleated stage at. So, as we have seen in the previous slide, that is micropylar end and chalazal end. So, at the micropylar end, egg apparatus is formed, out of which two synergid cell and one egg cell is present. Okay, in the central part that has a central cell. Now in egg apparatus, two synergy cells are present at the egg apparatus. A filiform apparatus which guides the entry of the pollen tube, okay, during fertilization. Now if we talk about the egg cell, which is haploid female gamete, okay, and formation of diploid zygote after fertilization takes place here. Now in the case of central cell, two haploid polar nuclei are present. Formation of triploid endosperm after double fertilization and triple fusion takes place. Okay. If we talk about the chalazal end, three haploid antipodal cells are present whose function are not known till yet, and it degenerates after fertilization. Now we are going to start with the next topic, that is pollination, which can be defined as transfer of pollen grains from anther to the stigma. Okay, which can be further classified into two types, that is self-pollination and cross-pollination. This self-pollination is further classified into two types, that is autogamy and geitonogamy, and cross-pollination is known as genogamy. Okay, the first one is autogamy, that is transfer of pollen grains from anther to the stigma of the same plant. That means the in this case autogamy takes place. Okay. Now, if we talk about the flowers, there are two types of flowers, that are chasmogamous and cristogamous flowers. Chasmogamous are the open flowers with exposed anther and stigma, whereas clistogamous are closed flowers. So coming to geitonogamy, transfer of pollen grain from anther of one flower to the stigma of another flower of the same plant is known as geitonogamy. Suppose, taking an example, this is a plant, this is the male flower, and here is the female flower. If the pollination is taken between these two flowers, it is said to be geitonogamy. Example of geitonogamy is cucumber. Now coming to genogamy, which is also known as cross-pollination, it is basically transfer of pollen grain from anther of one flower to the stigma of another flower that is born on two different plants. Suppose two different plants are here and the pollination is taking part between the flowers of these plants is known as cross-pollination. Now, pollinating agents can be abiotic agents or biotic agents. Now, abiotic agents can be wind or water. Wind pollination is known as aminophily, which is common in monocots like grass, whereas in case of water it's known as hydrophily, which is common in hydrilla, seawater. Coming to biotic agents, which is mostly done by animals by different ways. One of them is insect, which is most common pollinating agents, which is done by bees and butterfly. Other creatures such as reptiles, example garden lizard and lizard are also a pollinating agent. In case of birds, sunbirds and hummingbirds are pollinating agents, and in case of mammals, bat also acts as a pollinating agent. Coming to the features of flowers pollinated by different biotic and abiotic agents. Okay, so the first one is wind pollinated flowers. So in case of wind pollinated flowers, the pollen grains are non-sticky and light. They have well exposed anther and feathery stigma. They are also odorless, nectarless, and colorless flowers. They, are, they have small flowers that are present in bunches. When it comes to water pollinated flowers, mucilaginous coat is present on the floral parts. The most important point here is pollen grain are of ribbon shape or ribbon-like pollen grains are present. These flowers are also colorless, odorless, and nectarless flowers. Now, in case of insects pollinated flowers, these flowers have sticky pollen grains, attractive flowers with colorful petals, and flowers with edible pollen grains, and nectar is present at the base of the petals. Now, floral rewards for animals which are color, nectar, odor, and edible pollen grains, and the most important one is safe place for laying eggs. In case of Amorphophilous flower and yucca plant, the Amorphophilous flower is itself 6 feet height and which is a safe place for laying eggs. Okay, if we come up to yucca plants, then moth lay eggs in its ovarian cavity. An important note here, yucca and moth cannot complete their life cycle without each other. This is known as coexistence. Water is not a pollinating agent in all hydrophyte. Okay, in case of water hyacinth and water lily, we can see that it gets pollinated by insects or wind because their flowers are located above the water surface. Now we are going to see the advantages and the disadvantages of self and cross pollination. For the first, we are going to see the self pollination advantages, which are it maintains purity and there is no chance of mixing. Second, more chance of pollination. Then the third one is there is no need to produce large number of pollen grains. And the last one is no need of the floral rewards. Coming to disadvantages, it causes inbreeding depression, weaker progeny, and no variation, so it does not lead to evolution. Now, coming to cross pollination, the first one is advantages. So, advantages are higher yield, strong progeny. Progeny are highly resistant to several biotic and abiotic factors, and the last one is variation and evolution takes place. Disadvantages of cross pollination is appearance of some undesirable features along with desirable features, and the last one is uncertainty. Now coming to the topic outbreeding devices. in which plants have developed their own mechanism to prevent from inbreeding and to promote outbreeding. In case of monoecious plant, two types of flowers are there, bisexual and unisexual flowers. So in case of bisexual flowers, the time of release of pollen grain and stigma receptivity is different, so it generally protects autogamy. So it generally prevents autogamy, while the different way is the difference between the position of anther and the stigma. In case of unisexual flower, it prevents autogamy but not prevents gynogamy. In case of dioecious plant, it prevents both autogamy and zytogamy, as female flower is on different plant and male flower is on different plant. So basically this happens in the case of papaya. Coming to self-incompatibility, that is a genetic ability to recognize self-pollen grains and prevent pollen germination by the bisexual flowers. Pollen pistil interaction. So pollen pistil interaction includes all the events from landing of pollen grain on the stigma to the formation of pollen tube and entry of the pollen tube in one of the synergy cell of egg apparatus in the embryo sac. Now coming to the steps of pollen pistil interaction, which includes all the following steps. Now the first one is chemical interaction between the pollen grain and the stigma, as stigma should identify whether the pollen grain which has landed on the stigma is whether of the same species or not, right? Now here the pollen grain should be at 2-cell stage so that the further steps can be performed. Coming to third step, which is pollen tube formation. Now if all the things had happened in a right manner, then formation of pollen tube takes place through the style, and at the end the entry of pollen tube in the ovule of one of the synergy cell through micropyle end takes place. Now with the help of diagram we are going to understand this mechanism. Here the pollen grain, which is in 2-cell stage, is forming a pollen tube and reaching to one of the synergid cell, which is guided by pollen apparatus. Now we are going to start with the topic, that is artificial hybridization. Now the flowers has been classified into two types, right? That are bisexual flowers and unisexual flowers. In case of bisexual flowers, the male part, that is anther, has to be removed from the flower, that is known as emasculation. This process is known as emasculation, as the anther is removed from the flower. Now it is followed by bagging, in which the emasculated flower has to be covered with the butter paper so that any other pollen grain do not reach to the stigma. Now coming to the third step, that is artificial pollination. Now here the desirable pollen grain is dusted on the stigma after removing the butter paper. Now again the last step is rebagging. Once again the covering of a stigma with a desired pollen grain has to be done. Now if we come to unisexual flowers, as we all know that there is only one kind of reproductive parts in unisexual flowers, So there's no need of emasculation. We just need to do the bagging and then artificial pollination and rebagging in the similar manner with the bisexual flowers. With this, we came to the end of the first part of the video. Second part will be coming soon where we are going to complete the whole chapter. If there is any doubt, do ask me in the comment section. Till then, take care of yourself. Do like, share, subscribe to the channel. Thanks for watching. Keep on studying.